Hi all, today I'm going to discuss on the topic nitrogenous excretion. So here is a brief introduction on the nitrogenous excretion. So the nitrogen containing compounds when catabolized, they are producing the ammonia as a byproduct. So free ammonia is extremely toxic when it comes to the freshwater insect when they are inhabiting in an environment which is hypotonic to them. So the osmotic regulation which will be flexing the fresh water to pass through them, and it is easy to remove this toxic ammonia towards the fresh water. But in case of terrestrial insect, this is a problem to remove the ammonia as a byproduct in the waste matter. So in case of mammals and adult amphibians, they detoxify the ammonia by converting it into a soluble substance, that is a urea. So they are called as a ureotelic. And in case of birds, reptiles and insect, they are uricotelic. So they convert the ammonia into uric acid. So both are important. The both are terrestrial organisms which are converting to urea and uricotelic. The substance which are produced by the majority of the insect is a uric acid. That is the nitrogen. When it comes to the nitrogenous material, the 80% it will be uric acid. So the uric acid can be crystallized out easily and can be retained as a solid non-toxic waste substance. So it is important to remove the waste as a uric acid. So when we are discussing on the forms of nitrogenous waste, the first one is the ammonia. So it is a toxic waste and it can be excreted in significant amount in the moist environment or freshwater insect. So example, blowfly larvae and locust. And the second one is a uric acid, which will be the predominant nitrogenous waste seen in the terrestrial insect, because it is a low toxicity and the water conservation efficiency. We know that the terrestrial insect they should be in a water conserving mode, so that they will not be water deficient. So in case to conserve the water, they are releasing it as an uric acid. So example as orthopterans, coleopterans, lepidopterans, etc. And the urea is a third one, and it can be seen in some of the beetles. Example, nitrogenous beetle, etc. And allantoin and allantoic acid, which are the breakdown products of the uric acid, which are seen in significant amount of insects such as heteropterans and lepidopterans, like stick insect and Lucilia larvae. So the proportion when comes to the proportion of the these different types of nitrogenous waste, it will be different in different stages of the insect lifestyle. So in the life cycle, maybe the larvae will be producing one kind of nitrogenous waste, and the adult will be producing nitrogenous waste. That is the we can see the difference. അതായത് പല സ്റ്റേജസിലും പല ടൈപ്പ് ഓഫ് വേസ്റ്റ് ആയിരിക്കും പ്രൊഡ്യൂസ് ചെയ്യുന്നത്. The proportion of them will be different. So this is a table which shows the distribution of nitrogen in the excreta of insect. അതായത് ഒരു എക്സ്ക്രീറ്റയിൽ എത്ര എങ്ങനെയാണ് നൈട്രജൻ ഡിസ്ട്രിബ്യൂട്ട് ചെയ്തേക്കുന്നത്. അതായത് ഇഫ് ഇറ്റ്സ് യൂറിക് ആസിഡ് ഓർ അലൻറ്റോയിക് ആസിഡ്, യൂറിയ ആൻഡ് അമോണിയ. So this will be discussing further on our slides. So comes in next is the insects and their habitat. So first one is a terrestrial insect and the other one is a freshwater insect. When it comes to the terrestrial insect, they will be high in uric acid content. So we already talked about the water loss and all. So to minimize the water loss, it will be uric acid content will be more. In case of Schizocerca, the 55% uric acid and 40% it will be ammonia. And in case of the freshwater and moist environment insect, the ammonia can predominate due to the water abundance. So they can leave the ammonia. If it is toxic also, it is releasing in a large environment, that is a large water environment. So it is fine. So seen in the Aeshna larvae, in 74% it will be ammonia. So based on the different habitat, the proportion will be of the nitrogenous waste also will be differing. And some of the adaptations in excretion, this is just a few lines of the adaptations. That is, the environmental influence can lead to adaptation that can modify the nitrogen metabolism based on the diet and life stages. The second one is a storage excretion, that is they can retain their waste product as a uric acid uh, when it is a non-feeding stage, that is a pupation seen in the cockroach and desert locust. And enzymatic pathway is in the Drosophila. They lack certain enzyme but have other enzymes to generate urea and ammonia. So they are using an alternative pathway to generate the nitrogenous waste. So these are some of the adaptations which are seen in the excretion in some of the insect. Next is the nitrogen metabolism and uric acid formation. This is the main topic which I want to deal with, and I have written all these things in a few points so that it is easy to learn, and you can elaborate each and every sentence when you are writing an answer. So the first one is the source of nitrogen. Source of nitrogen means the nitrogen waste originates from a breakdown of protein and nucleic acid in the insect diet. അപ്പോൾ ഒരു പ്രോട്ടീനും ന്യൂക്ലിക് ആസിഡും ഡയറ്റിൽ ഉണ്ടെങ്കിലാണ് ഈ നൈട്രജനസ് വേസ്റ്റിന് സോഴ്സ് ഉണ്ടാവത്തുള്ളൂ. അമോണിയ is a byproduct of amino acid deamination, which is highly toxic and need to be efficiently managed. And second one is a conversion to uric acid. ഈ അമോണിയ പ്രൊഡ്യൂസ് ചെയ്ത് യൂറിക് ആസിഡായിട്ട് കൺവേർട്ട് ചെയ്യണം. So that can be detoxified and converted to uric acid by the purine metabolism pathway. And the key steps in that, the amino transferase and deamination. That is the first step. So the amino acid are deaminated to produce the ammonia, and the purine synthesis is the next step. So ammonia is incorporated into purine precursors such as inosine and monophosphate. The third one is the uric acid formation through enzymatic reaction. Purines are oxidized into uric acid and catalyzed by enzymes like xanthine oxidase. So they can catalyze into uric acid. And the third process is the transport to Malpighian tubules. After the conversion of uric acid, they will be transported to the Malpighian tubules. So in case of Malpighian tubules, we need to study about the role of hemolymph. We know that insect which are having hemolymph, which uric acid is transported in the hemolymph, that is the insect blood, from the metabolic tissue to the excretory organ, that is the Malpighian tubule. So through the hemolymph, this uric acid is transported to the Malpighian tubule and is a selective transporter. So the specialized epithelial cells of the Malpighian tubules actively transport the uric acid along with potassium ions into the lumen. So when we are talking about the role of the Malpighian tubules, they are the excretory structures which has a central role in the nitrogen metabolism, and uric acid is secreted into tubule lumen, often precipitated by VATPs. And in Drosophila, they lack this kind of enzymes, so they are using another we already discussed about the adaptation. They are using another enzymatic pathway to complete this process. And adaptive variation is a uric acid pump in the CC fly adjust activity based on urate levels in the hemolymph. And uric acid may be further metabolized to allantoin. We already said that it can be further metabolized to allantoin or allantoic acid in some species. And first we already discussed about the first few steps, and next one is the conversion to the secondary products. By the help of uric acid activity, uric acid is oxidized to allantoin by the enzyme uricase. And uricase activity is often restricted to the Malpighian tubules. That will be happening only in the Malpighian tubules and not in other areas. And allantoin and allantoic acid excretion seen in the Calliphora and Lepidoptera. The proportion of the uric acid, allantoin and allantoic acid depend on the diet and developmental stages. That also we already discussed, but if we are writing an answer, we need to write all these things. And developmental and species specific pattern, the balance of the excretory product shift across the life stages. That is Lasiocamba larvae, which excrete the allantoic acid on the grass diet but uric acid on birch. So these are the developmental and species specific patterns which we seen in the life stages of different insects. And next is the movement to the hindgut. After the Malpighian tubules, it will be moving to the hindgut, and they can be seen in the there is a reabsorption of water. The rectum, which are lined with specialized cells which can reabsorb water and essential ions from the excretory mixture, and this can ensure that the excretory product is concentrated, further conserving the water. Next one is the formation of excreta, which is a final composition will be resulting in excreta, which consist of uric acid, which is semi-solid or solid, depending on the insect species. And excretion, the excreta are expelled through the anus and completing the process of waste elimination. And here the significance of uric acid excretion is the water conservation, and second one is the detoxification. We all know that the excretion is for the detoxification, and environmental adaptation that can also go with the uric acid excretion. Next one is the uric acid storage and elimination. First one is the solid waste management. By the efficient storage, the precipitated waste can be stored temporarily in the hindgut without the osmotic disruption and the reduced water loss. It is also an adaptation we seen earlier. And excreta product and composition, that is the primary component will be uric acid, and the elimination which concentrate these components before ejection, and utilizing the rectal pads for the water reabsorption. So this shows that the terrestrial insect will be having minimal water ejection, so that the maximum water reabsorption will be possible. And the integration of the excretory system, there is a synergy between the Malpighian tubules and nephrocytes. That is, when the Malpighian tubules focus on uric acid production and osmoregulation, nephrocytes can handle other complex organic waste. The division of labor which can ensure the efficiency and adaptability in the insects. And role of fat body. The fat body serves as a metabolic hub. So it can contribute to the precursors for uric acid synthesis and integrating energy metabolism based on their excretory demand. So that is how the fat body works. So these can be also written as a small points in the essays. And filtration and secretion in the Malpighian tubules. The first one is the filtration. The hemolymph which have the nitrogenous waste and salt and small metabolites are actively transported to the lumen of the tubules. This active transportation of the potassium ions which can drive the osmotic water movement which can flow off the hemolymph in the tubules. And secretion, the nitrogenous waste is converted into uric acid which is insoluble and precipitate within the tubules. This solid state conversion minimizes the water usage during the excretion. That is how we require for the terrestrial insect. And here comes the role of nephrocytes. Nephrocytes are important in managing large waste molecules and the macromolecular absorption. Nephrocytes which absorb the high molecular waste substance that cannot be processed by the Malpighian tubules. So Malpighian tubulesന് പ്രോസസ്സ് ചെയ്യാൻ പറ്റാത്ത വേസ്റ്റുകളായിരിക്കും കൂടുതലും നെഫ്രോസൈറ്റ്സ് ഹാൻഡിൽ ചെയ്യുന്നത്. ദീസ് മോളിക്യൂൾസ് ആർ ഡിഗ്രേഡഡ് ഇൻട്രാസെല്ലുലർലി ആൻഡ് ദ ബൈ പ്രൊഡക്ട്സ് are either recycled into the hemolymph or directed to the Malpighian tubules for the excretion. Now we are talking on the synthesis of uric acid. There are different mechanisms for the synthesis of uric acid. The first one is we are dealing with the de novo synthesis. So starting point is the 5-phosphorybosyl 1-pyrophosphate, which is PRPP as a precursor, which combines with the amino acid and formate to form the purine ring. And the second process is the building of the inosine monophosphate, that is the IMP, through a series of enzymatic reaction, that is using the glycine, glutamine and aspartate which can contribute to the nitrogen and carbon to the purine ring. And IMP is formed by a central molecule which serving as a precursor for other purine derivatives. And the third process is the conversion of IMP to uric acid, and this is the final step. And IMP will be converted to inosine, and inosine to the hypoxanthine, and hypoxanthine to the xanthine, and xanthine to the uric acid. So these all can be done through the many enzymatic reactions, and these are explained here. That is the inosine formation is undergoing the dephosphorylation and loses its ribose sugar, and hypoxanthine formation is a sugar removal which lead to the hypoxanthine formation, and xanthine oxidase convert the hypoxanthine into xanthine, and finally it will be oxidized to form the uric acid. സോ ഈ ഡിനോവോ സിന്തസിസ് എന്ന് പറയുന്നത് വളരെ ഷോർട്ട് പോയിൻറ്റ്സ് ആയിട്ട് എഴുതിയിരിക്കുന്നത്. അത് എക്സ്പാൻഡ് ചെയ്തിട്ട് നെക്സ്റ്റ് സ്ലൈഡിലൊരു പിക്ചർ കൂടി ഉണ്ട്. അപ്പോൾ ഒരു 15 മാർക്സ് അല്ലെങ്കിൽ 25 എസ് എ ആയിട്ട് ഈ യൂറിക് ആസിഡ് സിന്തസിസ് ഒക്കെ ചോദിക്കുകയാണെങ്കിൽ ഇതും വേറൊരു പ്രോസസ്സും ഉണ്ട്. അത് രണ്ടും കൂടെ കമ്പൈൻ ചെയ്തിട്ട് എഴുതാം. സോ യു ക്യാൻ ഓൾസോ റൈറ്റ് ദീസ് ടേബിൾ ടു the answer if it is an essay question. സോ ഏതർ നിങ്ങൾക്കത് വരച്ചിട്ട് എഴുതാം, അല്ലെങ്കിൽ ഒരു ഫ്ലോ ചാർട്ട് പോലെ യു ക്യാൻ റൈറ്റ് ദ നെയിംസ് ഓഫ് ദീസ് തിങ്സ്. ആൻഡ് ദ നെക്സ്റ്റ് ആക്സെപ്റ്റഡ് ആയിട്ടുള്ളൊരു മെക്കാനിസം എന്ന് പറയുന്നത് യൂറിക് ആസിഡ് സിന്തസിസിൻ്റെ ന്യൂക്ലിക്കോലൈറ്റിക് പാത്വേ ആണ്. അത് ന്യൂക്ലിക് ആസിഡ് വെച്ചിട്ടാണ് ഈ ന്യൂക്ലിക്കോലൈറ്റിക് ആയിട്ടുള്ള പാത്വേ അണ്ടർഗോ ചെയ്യുന്നത്. So when we are comparing with the uricotelic pathway, this pathway is simple and it is rather small pathway, and the energy conservation reaction where the breakdown and destruction of it can be kept in control and their activities are regulated by the nucleolytic pathway. And here is the nucleolytic pathway. First one is the degradation of the nucleic acid. The nucleic acids are broken into nucleotides which are further hydrolyzed into nucleosides, that is the adenosine and guanosine, and the formation of the free purines by the help of enzymes adenase and guanase which is deaminate nucleosides into free purines such as hypoxanthine and guanine. And guanine is further converted into xanthine, that is the hypoxanthine is converted into xanthine again, and the xanthine oxidation which will be leading to the uric acid. So as mentioned earlier, it is a small pathway when comparing to the uricotelic pathway. So this can there is also a flow chart which can be used for the essay question. And the next topic is, and final topic is, a storage excretion. The ways we already discussed about the storage excretion before, since we are already seen in the cockroaches and all. So here we are detailing about the storage excretion further. So it is the waste materials which are retained in the body. അതായത് നമ്മുടെ ബോഡിയിൽ ആ വേസ്റ്റ് മെറ്റീരിയൽസ് കുറച്ചും കൂടി നാൾ സ്റ്റോറേജ് വയ്ക്കുന്ന ഒരു മെത്തേഡാണ് ഈ സ്റ്റോറേജ് എക്സ്ക്രീഷൻ എന്ന് പറയുന്നത്. അപ്പോൾ യൂറിക് ആസിഡ് ഇൻസോല്യൂബിൾ ആയതുകൊണ്ട് തന്നെ ദേ ക്യാൻ ബി അവൈലബിൾ ഇൻ എ സോളിഡ്. സോ ഇറ്റ് ക്യാൻ ബി സീൻ ഇൻ ദ ഫാറ്റ് ബോഡി ഓഫ് മെനി ഓഫ് ദ ഇൻസെക്ട് ഇൻ ദെയർ ഡിഫറെൻറ്റ് സ്റ്റേജസ് ഓഫ് ഡെവലപ്മെൻറ്റ്. അതാണ് നമ്മൾ കോക്രോച്ചിൻ്റെ പ്യൂപ്പിലൊക്കെ കൂടുതലും കണ്ടുവരുന്നു എന്ന് നേരത്തെ പറഞ്ഞത്. So sometimes the uric acid and other nitrogen compound which can be permanently deposited in the epidermis so that it can lead to the color pattern of the insect. ചില ഇൻസെക്റ്റിൻ്റെ ഓരോ കളർ പാറ്റേൺസിന് പോലും ഈ യൂറിക് ആസിഡിൻ്റെയോ അല്ലെങ്കിൽ നൈട്രോജനസ് കോമ്പൗണ്ട്സിൻ്റെ സ്റ്റോറേജ്, അതായത് ബോഡിയിൽ സർട്ടൻ പാർട്സിലുള്ള സ്റ്റോറേജ് ലീഡ് ചെയ്യുന്നുണ്ട്. അതായത് അവരുടെ കളർ പാറ്റേൺ അല്ലെങ്കിൽ ആ ഡിസ്റ്റിങ്വിഷ് ചെയ്യുന്ന കളർ പാറ്റേണിലോട്ട് പോലും ഈ സ്റ്റോറേജ് ലീഡ് ചെയ്യുന്നുണ്ട്. ലൈക്ക് വി ക്യാൻ സീൻ ഇൻ ദ white markings in the larval instars of the cotton stainer bug, and this can be result from the accumulation of the uric acid. Also in the white color of the cabbage white butterflies, it is due to the pterins which represent the end product of the nitrogen metabolism and can account for the 14%. അതായത് 14%ഉം ഈ ടെറിനാണ് നൈട്രോജൻ മെറ്റബോളിസത്തിൻ്റെ വേസ്റ്റ് പ്രൊഡക്റ്റ് ആയിട്ട് നിൽക്കുന്നത്. സോ അത് കാരണം ഒരു വൈറ്റ് കളർ cabbage white butterflies we can also see. And compounds which other than the nitrogenous waste are that is the calcium which can be stored like this as a along with the uric acid. Calcium which can be accumulated can increasing on the successive stages of the development, and it can be in higher concentration of calcium which can be accumulated in the tubular regions of the midgut. Also the calcium is stored in specialized regions of the malpighian tubules, that is for example in larvae of the fly which are living in the water containing high levels of calcium salt, which will be storing the calcium in enlarged malpighian tubules. Also seen in the stick insect, the terminal regions of the inferior tubules are expanded to store the calcium. Also we can see the heavy metal accumulation in the midguts of the caterpillar and the posterior midgut cells of the larvae of the midge and etc. സോ ഹെവി മെറ്റൽ അക്യുമുലേഷനും കാൽഷ്യത്തിൻ്റെ അക്യുമുലേഷനും അതുപോലെ തന്നെ യൂറിക് ആസിഡിൻ്റെ അക്യുമുലേഷനും ഇതൊക്കെ നമുക്ക് ഓരോ ഇൻസെക്ട്സിലും ഓരോ സ്പീഷീസ് അനുസരിച്ച് നമുക്ക് പ്രത്യേകതയിൽ കാണാനായിട്ട് പറ്റും. ആൻഡ് ദേ ക്യാൻ ബി ലീഡിങ് ടു സം ഓഫ് ദ മോർഫോളജിക്കൽ ക്യാരക്ടേഴ്സ്, അതായത് വൈറ്റ് മാർക്കിങ്സ് ആസ് വി സീൻ ഇൻ ദ സം ഓഫ് ദ കാറ്റർപില്ലേഴ്സ്, അത് വൈറ്റ് മാർക്കിംഗ് ഓൾസോ ഇൻ ദ ക്യാബേജ് വൈറ്റ് ബട്ടർഫ്ലൈയിൽ വൈറ്റ് കളറേഷൻ ഒക്കെ ഈ വേസ്റ്റ് മെറ്റീരിയൽസ് കാരണം ഉണ്ടാവുന്ന കുറച്ച് മാർക്കിങ്സ് ഒക്കെയാണ്. So this is all about the storage excretion. Here is a few books and site I referred for preparing this. Thank you all for listening.